Hello, today's video is entitled Calcium Alpha Ketoglutarate or calcium AKG, TET enzymes, and our epigenome. What is our epigenome? Okay, not rather than genome. The epigenome is the complete set of chemical switches. I mean, switch on or switch off your gene. You switch on or switch off. Or modifications on your DNA and its associated proteins called histones, which I'll explain what they are, that control which genes are turned on or turned off, essentially acting as a software for your genetic hardware, your genome. So that means that your genes is the hardware, but the epigenome is the software. I mean, you may have genes from your parents that you are prone to diabetes. But because you live healthily, because you take certain supplements and all that, your epigenome will turn off those bad genes, and they can turn on the good genes, right? So these marks like DNA methylation and histone modification, they don't change your DNA code itself. I mean, your DNA, half is from your father, half is from your mother. Your DNA doesn't change, but it just changes the expression, which one are expressed, which one are turned off, which one are turned on, which one are turned off. But they alter the gene activity, determining cell type, liver or blood cell, and responding dynamically to development, diet, stress, and disease. That means which genes are turned on, turned off, which good genes are turned on, which bad genes are turned off is depending on your lifestyle, right? And they also determine the cell type when you're early in the embryo stage. Like your liver cell, your skin cell have the same DNA, right? Same DNA that's inherited from your parents. But why is this liver cell? No, why is this skin cell and this is liver cell? Because the DNA here in the skin cell, they say turn off the liver gene. Don't express the liver gene here. You are a skin cell, right? That's basically what the epigenome does. Okay, to explain the epigenome further, there are chemical marks. It's a collection of tags, including methyl groups, added to the DNA. When you methyl something part, it's called methylation. That means the gene is turned off. If demethyl, take away the methyl group, the gene is turned on. Okay. And modification to histone proteins that package DNA, influencing how tightly the DNA is wound. I mean, see, our DNA is like in coils, right? In coils. So if the proteins are hidden because it's wounded, that means it cannot be expressed, right? So all these things I'll explain. Gene regulation. These marks dictate when, where, and how much a gene is expressed, enabling a single genome to create diverse cells and function. Dynamic and responsive. Unlike static DNA sequence, that means your genetic material, your genome, the epigenome changes throughout life. Right? It's influenced by environment experiences like maternal diet and pregnancy, and aging, and exercise, and diet, right? Cellular identity. It's crucial for cellular differentiation, ensuring a skin cell behaves differently from a brain cell, even though they share the same DNA. Remember, I said all the cells in our body has the same DNA, but your brain cell will express the brain genes, right, and turn off all the other genes because it's told to be a brain cell and not a liver cell or a heart cell. Health and disease. Epigenetic patterns differ between healthy and disease states. Like you have cancer, the epigenetic has changed, so allowing it to be cancer, and can be even passed on transgenerational inheritance, making a key area in medicine. That means if you are not having a healthy lifestyle, and then your epigenetics are no good, right? Your software is no good because you are drinking, smoking, whatever, then you can even pass it on to your kids, right? So you have to be very careful. So in essence, if the genome, I mean, is the hardware, your genes, the epigenome is the operating system, managing and interpreting the instructions for life, okay? It's just like your computer is the hardware, but the software, like the Windows, DOS, whatever, is the software. Okay, what is epigenetics? I'm sure you all heard of people go for a DNA test, right? But you don't even know what you're testing for, how it works. like take from your blood or take from your saliva, a swab, then it's going to go for gene testing. It's actually epigenetic testing to see your risk profile for certain diseases, whether you're high risk for cancer, for breast cancer, for colon cancer, for diabetes. So they take a bit of your DNA. So epigenetics is the study of how behavior and environment can cause changes that affect the way your genes work, your epigenetic profile, right? Your epigenome. Unlike genetic changes, epigenetic changes are reversible and do not change your DNA sequence, but they can change how your body reads, right, and express a DNA sequence. Gene expression refers to how often or when proteins are created from instructions within your gene. I mean, your gene has all the hardware, all the coding, what to do, how to live, how to make enzymes, how to burn fat, how to detox, right? It's all in your genes, right? But then you need proteins. Then the gene need to make proteins to actually function, to actually do the effect. I mean, carry out the work in the body, in each organ, in each cell, right? While genetic changes can alter which protein is made, epigenetic changes affect gene expression to turn certain genes on or turn certain genes off. Okay, so hopefully, if you have a good lifestyle, you turn off the bad genes and turn on the good genes. So, since your environment and behavior such as diet and exercise can result in epigenetic changes, it is easy to see the connection between your genes and your behavior and the environment. How does epigenetics work? Epigenetic changes affect gene expression in different ways. Types of epigenetic changes include DNA methylation. DNA methylation, which I mentioned, works by adding a chemical group called methyl group to DNA. Typically, this group is added to specific places along the DNA where it blocks the proteins that attach to DNA to read the gene. That means that when you put a methyl group, the proteins cannot read the gene, so you cannot express that gene, right? This chemical group can be removed through a process called demethylation. So you remove the methyl group, then the gene is turned on, right? When you methyl it, then the gene is turned off. So another way how epigenetics work is histone modifications, okay? DNA wraps around proteins called histones. I mean, DNA is like a long shoelace, isn't it? But it is wrapped around proteins called histones. Wrap, then they go to another one, wrap, then go another one, wrap. When histones are tightly packed together, proteins that read the gene cannot access the DNA easily, so the gene is turned off, isn't it? When it's all packed, the proteins cannot read the DNA. When histones are loosely packed, more DNA is exposed and not wrapped around a histone and can be accessed by proteins to read the gene, so the gene is turned on, right? Chemical groups can be added or removed from histones to make histones more tightly or loosely packed, turning genes on or turning genes off.
Another epigenetic process is histone modification. The long coils of DNA in our cells are wrapped around clusters of proteins called histones. How loosely or tightly they're wound around these histones controls how little or how much of the gene can be read and expressed.
So, while DNA methylation is...
Okay, TET enzymes, okay, roles have come to light only in the last 5 years. They are needed for stem cells to maintain their multipotent state and are involved in early embryonic and brain development in cancer, right? So, for instance, here you go. TET enzymes in epigenetic regulation in a mammalian neurobiology. Important, for embryonic neurogenesis, postnatal brain development, adult behavior, human disease. I won't go too far into it, but it's very important for our epigenetics. Okay, TET enzymes, okay? Using oxygen, that means they use oxygen when you breathe. TET enzymes, see here, 1, 2, 3, change 5-methyl cytosine here, 5-MC, right? And other oxidized forms of methyl cytosine, okay? It's all starts with cytosine, so it's very important for intake. Into other oxidized forms of methyl cytosine, 5-MC and 5-HMC. Here you go, the two, and other forms of MC. Okay, why are these important? Because they are both epigenetic modifications of DNA, which change how DNA is regulated without altering the letters of genetic code itself. So TET enzymes are very important for how your genes are expressed. TET enzymes, for instance, 5-MC are generally found on genes that are turned off or on repetitive regions of the genome. 5-MC helps shut off genes that aren't supposed to be turned on, right, depending on the cell type. And changes in 5-MC distribution underpin a healthy cell's transformation into a cancer cell. That means if your 5-MC is not working, the genes that are not meant to be turned on is turned on because there's not enough 5-MC. Then it can lead to a cancer cell. Alterations of TET enzymes have been found in forms of leukemia, so having information on the enzyme's molecular structure could help scientists design drugs that interfere with the process of cancer development. So, AKG and TET proteins, alpha-ketoglutarate. Alpha-ketoglutarate and TET proteins are crucial partners in epigenetics, where AKG acts as a vital cofactor for TET enzymes, like TET 1, 2, and 3. See, here's TET 1, TET 2, TET 3 enzymes. And you need AKG as a cofactor for the enzymes to work, right? Because these enzymes initiate DNA demethylation by converting 5-MC to 5-HMC, driving epigenetic reprogramming in development, cell differentiation, and disease, with AKG levels impacting TET function and overall epigenetic health. So if your AKG is low, your TET enzymes cannot function, and then it will lead to certain health conditions. Okay, a lot of people have been taking 4th generation nutritional supplements like NMN, which I was taking also. But now I'm also taking calcium alpha ketoglutarate, AKG. What's the difference between the 4th and 5th generation? Is that the 5th generation alpha ketoglutarate can influence your epigenetics, genetic balance, right? So it can influence your epigenetic balance for the better, the expression of your epigenetics, which the 4th generation supplements like NMN cannot do. There are many types of AKG in the market, but the L-arginine AKG is essentially for bodybuilders anyway. So essentially when you talk about epigenetics and AKG, it's calcium AKG, which we refer to. Okay, AKG TET enzymes connection. TET enzymes, the key players, TET actually stands for 10-11 translocation proteins, are a family of enzymes. They are alpha-ketoglutarate dependent dioxygenases, meaning they need AKG to function. Here you go, AKG dependent demethylation, to remove the methyl group for genes to express, to turn on. DNA demethylation pathway. These oxidized forms are intermediates in removing methyl groups from DNA, a key genetic reset process that leads to gene activation or reprogramming, crucial for development and stem cell function. So for your stem cells to function, you need to demethyl. Why it matters? What's the epigenetic of low AKG? Right? What's the epigenetic impact? TET and AKG are vital for resetting DNA methylation patterns in early embryo and maintaining stem cell pluripotency. So if you are pregnant, if you have low AKG, it will affect your embryo, the development of your baby. Cell differentiation. Regulating TET activity helps control cell fate, which with TET deficiency linked to block differentiation. Disease. Dysfunctional TETs, often due to low AKG or mutations, are implicated in cancers like leukemia and metabolic conditions like diabetes, impacting gene expression and causing disease. Metabolic link. AKG levels can fluctuate with diet and metabolism. Low AKG, for example, with people with diabetes, impairs TET function, disrupting the epigenome. In essence, AKG fuels the TET machinery, which controls DNA methylation, making it a critical metabolic link to epigenetic regulation, influencing everything from cellular identity and to disease susceptibility. So if your AKG is low, your TET machine is not working, your TET enzyme is not working, you're more susceptible, more prone, more at risk of diseases. So I hope that was helpful. Thank you for listening.